What's up, Baby Bio Penguins? So today we are going to do a Unit 8 review. We're going to talk about ecology, and everyone has their favorite unit, and well, this is not mine. But hey, we are going to rock through this because we are amazing and we are penguins and we are ready to rock this. So first thing, you are sitting in my live, which means that you are here, you are ready, you are dressed to success. So you are now part of my grouping of penguins. I hope you are excited to be part of my huddle. So what different resources do we have? Okay, so we have 9 days for the AP exam. So like all of these resources might not be the most, like what you got to do all of this. But so I've got daily review on my Instagram stories. At this point, I have ended the review, and I'm doing the 2013 exam next week, and then I'm just doing Q&A. So as students are asking questions, I'm answering them. I'm only one person, so I do my best. But I think this morning I answered like 30 or 40 different questions on there. So if you got questions, you can always ask them on my Instagram story, and I'll do my best to respond to you. Okay. I have a 505-page review guide. Now, at this point, you can't do the whole 505 pages, but you can go through it and you can find the multi-choice questions for each of the units and you work through those. You can find certain FRQ questions from each of the units, and that would kind of help you to fine tune a little bit of your studies. Okay. I've also got recorded FRQ Fridays. So every single FRQ from 2013 to 2025, yes, I finished the last one yesterday, is all available on YouTube for you. So you can go back to any of those and kind of hear someone talk through the question, which might be helpful for you. Plus, I can show you tips and tricks about how to get points when you don't know anything. We also have 120 Wayground games. So there is, all these review games. You can go in there and just do quick review on any of the information. Okay. As well as I've got all these review videos and review PowerPoints. So everything is on my website, apbiopenguins.weebly.com. And I'm noticing there's a lot of stuff happening in the chat. I'm one person, and if you see an answer to a question, you're welcome to answer each other. There is a slight delay between when you write it and when I see it. And if I'm like in the middle of explaining something, I'm going to finish what I'm explaining, and then I'll go back and answer your question. So if y'all see each other's questions and you want to help each other. So Unit 8 is 10 to 15% of the exam. And so it's not like a huge chunk, but we do find that there's a lot of other questions that are kind of nestled in ecological questions. So we're going to look at response to the environment and like animal communication. We're going to look at energy flow and the nutrient cycles because they just got added back into the curriculum. We've got population ecology, community ecology, and then any disruptions that are happening to the ecosystem. And then we'll work through a couple practice questions, and then we'll talk about the exam format and other additional resources that might help you. So let's just get into it. Okay, response to the environment. Okay, so organisms have to be able to respond to their environment, and then so they're going to look at different internal and external cues. And so as they do that, that can cause changes in their behavior. So there's a lot of different types of communication. Sorry, I'm still got this cough. So we have visual. Visual, of course, is things that you can see. Okay. So fireflies are going to glow, and by glowing, they can attract themselves a mate. We also see our peacock is going to show off their, beautiful tails when they're going through their courting rituals. And then the cobras are going to inflate their hood and kind of scare off other organisms. Okay. So those are all visible, kind of visual, like cues that might come from the environment. Okay. We also have auditory. Auditory is going to be things that you can hear. Okay. And so the elephants are going to use their trunks to talk to the other herds that are long distances. We know we have echolocation where we can look at male whales might also use their song to communicate with the females. And then wolves are going to howl in order to kind of talk to other wolves that are in the pack. Okay. So we have tactile. Tactile has to do with touch. So dogs are going to lick their pups in order to kind of bond with them. Baboons are going to touch one another to kind of show affection and groom. And the horses oftentimes kick other horses to establish their dominance. And then the last one we also have electrical. Electrical just has to do with the simulations. And then chemical has to do with the different materials that are released out. So like cats are going to rub against different objects so they can mark them with their scent. We also know that individuals are going to like pee and urinate on different things. Ants have a pheromone trail. So if you've ever got ants and you want to get rid of the ants, just get rid of the pheromone trail and they'll stop following each other. And then skunks are going to use different signature smells to deter different predators. Okay. Now, on the exam, what's probably going to happen is they're probably going to give you an example and you're going to apply information to it, or they'll tell you about some type of communication and you're going to then answer something about it. They're not just going to willy-nilly ask you for a type of communication. Okay. So the reason why we have to go through communication has to do with that we want to indicate dominance. As we said before, we're going to indicate the horses are going to kick one another to show their dominance. And we might see where two deer are going to kind of hit each other, just trying to like show which one is going to be dominant in the group. Okay. Also for foraging food, we got to be able to find food, and if they can't find food, they're going to have problems. Okay. So ants oftentimes when they leave the oh my god, the hill, they are going to rub their antenna against the one that's coming in, and that's how they communicate with each other about like where the food is. The bees are going to do a waggle dance so that they can tell where the food is. They, based on the length of the waggle and the angle of the waggle, it tells the other bees where to go to get the nectar. Okay. They also do to establish territory. So oftentimes we might see where a bird is going to have to get a nest, and based on that nest is going to not have any others in the area. So it marks territorialism as well as marking its area so they can, of course, ensure reproductive success. So response that we have to the environment. Okay, so what you see right here, this is going to be kinesis. So this is a sow bug, or you may know them as roly-polies. Yeah, tactile is physical contact. So this is a roly-poly, also known as a pill bug. And so what it happens is that it is going to move quicker when we're going to find that it's in a dry, arid environment, like that's just dry, it's, not happy. Okay. Versus once it's underneath like this leaf, it's going to kind of be in a moist environment. It's kind of dark, and that's kind of favorable for it. And so it's going to not move as fast once it is underneath the leaf. It has no way to sense where the leaf is to then say, I'm going to move to the leaf, which is why you see it kind of moving all over the place. So that's an example of kinesis. And so based on the environment, it's going to change their behavior. If it's in a dry environment, it moves fast. If it's in a moist environment, it moves slow. So we also have intersexual selection. Okay, so intersexual selection has to do with where one sex is choosing the other sex. Electrical could be like an eel, like an electric eel might release electrical signals out. So reproductive barriers. Okay, so what's going to happen is that, like, so our blue-footed booby. The blue-footed booby is going to show off his little dance, okay? It shows off its feet. And so that's going to allow it to attract a mate. So we're going to see where one is going to be choosy of the other. So it's like inter, like within, one versus the other, kind of choosing one another. We also see that frogs can kind of make their mating noises to try to attract the other mate. Versus intrasexual. Yeah, I know, I scared you on that one. Intrasexual has to do with that they're going to indicate that dominance, and they're going to compete for access to that mate. Okay, so here you can see these two are going to fight so that they can be the alpha male, so they can have access to mating with all the others. Okay, you also see that these little ants over here are like, they're fighting also for a mate. And so with this one, it's going to be two of the same sex that's going to kind of fight for the other. Okay, so different responses that we have for our environment. Okay, so fitness favors both our innate and our learned behaviors. Okay, so innate is going to be something that you were born knowing how to do. Like you were already born, knowing how to do this. Versus learned behavior is something that, due to trial and error during experiences, you've learned how to do something. Okay, and so altruistic behavior is going to deal with that. We're going to look at that. The individual fitness is going to include fitness. The best example I can come up with is belly squirrels. Okay, so with the belly squirrels, what's going to happen is they're going to let out an alarm call if they see a predator. Okay, and so because of that alarm call, what's going to happen is the predator is going to know exactly where that squirrel that made the alarm call is, and is now going to go after that squirrel and probably get it. Okay, but by making that noise, by letting out that alarm call, it tells all the other squirrels to hide. Okay. Now, the reason why it does this is due to kin selection. Okay. They are very closely related to each other. And so by letting out that alarm call and telling the others, hey, there's danger, it's, protecting its own genetic material in a way. Okay. Where it's ensuring that like its genes will continue going on to future generations because that predator didn't come in and get all of the squirrelies. Okay. And so we also look at cooperative behavior, where we're going to increase the fitness of the individual and the survival of the whole population. So I was like, okay, I need some examples of this. And so behavioral responses would be things that are different within your behavior, and psychological, I'm assuming, or have to do with like the function. So of the actual response, you're looking at the functionality of it. So I looked at examples this morning of cooperative behavior. So the different examples we saw were cooperative hunting. So like they might hunt in packs, okay? And so by hunting in that pack, they're going to be able to use teamwork. And so that's some little bit of cooperation right there. We also see cooperative breeding. And so two are going to, of course, breed and they have their young, but then the other individuals within the population are going to help them with those young. They'll do different babysitting skills. They'll help them get food. They'll help raise them. And so that's cooperative breeding. And so again, they're working together. And then cooperative defense, that was what I was just mentioning about how we have the altruistic behavior where one of them might call out a signal, which then helps the whole population to survive. We also saw where we could have mobbing behavior, and mobbing behavior has to do with that they're going to harass, which can help to drive away a certain predator. Okay, so looking at ecosystems, okay, so now we look at the body temperature. Okay, so there's two different types of organisms we have. We have endotherms and ectotherms. Endotherms are going to be what you call a warm-blooded organism. Now, we're not calling them that because we are in AP biology, but endotherms are going to be the ones that go through metabolism. And going through metabolism is going to allow them to generate the heat to keep their bodies warm. Okay, so yes, they all go through metabolism, don't get me wrong there, but they are going to use their metabolism to keep themselves warm. So if you notice here, no matter what the outside temperature is, the internal temperature of this organism is always going to be the same approximate temperature. Because when it's colder, it's probably going to respirate more frequently to maintain that higher temperature. And so oftentimes they might look at a question where they'll look at like oxygen consumption. And we would see that there would be a higher oxygen consumption at a lower temperature because they're going through metabolism more frequently. And we all know that the final electron acceptor is, of course, oxygen. So you would consume more oxygen when it's in a colder environment as an endotherm. Versus an ectotherm, it's going to use different behaviors to maintain its temperature. Okay, so it's going to go bask in the sun. It's going to go kind of cuddle together with others. It's going to burrow. It's going to do different things so that it uses the environment to keep itself warmer or cooler. Okay, and so it oftentimes is going to match the environment. And so here we see if it's cold outside, it's cold inside. If it's warm outside, it's warm inside. And so it actually is a conformer, and it conforms to whatever its environment is. Versus the endotherms are regulators, and they're going to have a different temperature. Okay, and so if we were looking at two different organisms and it was cold, which one would metabolize more frequently? It would of course be your endotherms. Also thinking about like snakes and why do they only have to eat once a week? Because the fact that they're not really using that food to keep themselves warm, and so they can kind of chill for a little bit long. Ecto is the ones that are going to kind of bring in external environment temperature, versus the endo is within itself. So in terms of the energy flow, we need to have a net gain of energy. Okay, so when there's a net gain, it's going to store that energy up. It's going to allow the organism to grow. And then we're going to see an increase in their reproductive output. Because there is more food, because there's more energy, these organisms are going to have larger groupings and larger amounts of offspring because there is enough to supply for them. Versus if we have a net loss of the energy, of course, we lose mass, we're going to see a decrease in reproductive output. So whenever there is a drought or whatnot, you might see, like, let's say a bird, right? If the bird normally has 8 young each of the times that it reproduces, maybe it's only going to have 4. And it has to do with because of the fact that they're decreasing the reproductive output because there's not enough resources for them. Okay. And then also, if there's not enough energy, we'll see death to the actual organism. And now, this sometimes I read the CED and I find information that I've never known before. And so this is one of those things that I didn't know until I was reading through the CED. And so the CED says that different organisms are going to use reproductive strategies in response to energy availability, and they can alternate between asexual and sexual reproduction in response to their energy availability. And I didn't know that was even possible, but apparently there are. There's lots of organisms out there. But this is one example. It's an aphid, okay? And so in the spring and the summer, what's going to happen is going to go through parthenogenesis. Parthenogenesis is basically mitosis, if you may. It's asexual reproduction, okay? And so they'll reproduce a whole bunch of these offspring. There are a whole bunch of the aphids, right? And then once it starts to get colder, okay? The days get shorter and it gets colder. They're going to go into sexual reproduction, and they're going to form these little eggs. And because of the fact that they can survive through the winter, okay, they're okay in that cold temperature because, again, we're looking at insects. Insects are going to be ectotherms. They're not going to do so good in the cold temperatures, okay? And so they're going to alternate between being asexual in certain times of the year and sexual in other times of the year. Isn't that so cool? Okay. Anyway, so energy flow through the ecosystem. So we talk about our different individuals, and then a bunch of the individuals that are of one species can make up a population. That population, of course, is the same species, okay? Now, within the area, you might have a community. That is multiple different species in that same area. And then the ecosystem is looking at the abiotic and the biotic factors within that same area. And so we look at the energy, right? So light is where all the energy comes in. So light shines down. The light energy is going to bring into the sun is going to bring those photons, all that energy into our primary producers. They're going to absorb that energy, and then they're going to go through photosynthesis, which is then going to allow them to create the biomass that allows them to, of course, make food, right? And then they're consumed by our primary consumers, who are consumed by our secondary consumers, who are consumed by the tertiary and the quaternary. We have that 10% rule, okay? Only 10% gets passed to the next generation. So of the 20,000 kilocalories that we have here, there's only 2,000 kilocalories, and then 200 kilocalories, and then 220, and then 2, okay? So we only have 10% moving on to the next generation. This one's a little bit different because we're looking at producers to our consumers. But once we get into that consumer level, we'll look at 10%. Now, and every single one of these, we're going to lose energy in the form of heat, okay? And so heat is constantly coming out. And then in terms of the kind of reusing of our resources, the decomposers are what's going to kind of help us to regenerate and reuse our different like nutrients. But if we're looking just at energy, energy is going to come in and then come out. They might ask you to calculate. There has been questions where they've given you the numbers in a chart, and you've had to figure out, what was the amount of energy that went from one generation to the next, or what fraction of the energy. There was a numeric response about that a couple of years ago, but don't worry, there's no numeric responses now. They're just embedded in the free responses, embedded in the multiple choice. So trophic structure, we have our autotrophs. What does 10% mean? 10% means that 10% of the energy goes to the next level. So autotrophs, these are going to capture the energy in the form of some type of physical or a chemical source. Physical being sunlight, chemical being different chemicals. And so if we're looking at sunlight, that would be something that is photosynthetic. If it is using chemicals, it's chemosynthetic, okay? And so chemosynthetic is going to be using small inorganic molecules in our environment. So thinking about like down at the heat sink, you've got a bunch of different bacteria that live down there, and those are going to go through chemisynthesis, and they're going to actually be able to make food for the other organisms in that area. And that's how that like deep sea area is able to have its own little ecosystem, is because they're generating from the heat and from the molecules that are coming in from those deep sea vents. So then we have our heterotrophs. Heterotrophs are going to get energy from something else, okay? And so they're going to use different carbon compounds that were produced by other organisms, and then they're going to consume their organic material and then incorporate it into their own tissues. They will metabolize carbohydrates, lipids, and proteins. Notice that it doesn't have our nucleic acids, okay? So nucleic acids are never used for metabolism. They are consumed, they're broken down, but we just reuse those nucleotides to generate and make more DNA or RNA, because we use those individual nucleotides to build the next component, okay? And again, they're going to use hydrolysis, which would be the breaking of the bonds, okay? So the energy goes from the autotroph to our heterotroph. Now, someone says, what does trophic mean? Trophic is just talking about the different levels we have, okay? So this would be our trophic structure, is looking at the different levels within that ecosystem. Okay, now based on availability of resources, we might see slight differences, okay? So if there is a change in the amount of energy, that's going to affect the size and the number of organisms that are in our trophic structure, okay? Because of the fact that if we see a rapid decrease in the primary level, well, there's not going to be enough food and not enough energy to support the next level. And I think I just jumped into the second one. I'm sorry. Affects the number and the size of the trophic levels. Anyways, but both of these are looking at the amount of resources going up. Okay, so if you don't have enough resources at the bottom level, then the next level is not going to have enough either. And so it'll be smaller and smaller and smaller. It will kind of see a decreasing as we go up. And then same thing with our decreasing of biomass or the decreasing of our producers. One of the examples we're going to look at actually dives into that. And so energy gets, it flows through a system. But our matter and our nutrients, they're going to cycle. So we have our biogeochemical cycles, okay? This is going to help us to demonstrate our conservation of matter, and they're interdependent, so they don't actually kind of, whatnot, okay? Now, abiotic and biotic reservoirs, okay? So there's going to be both living and non-living components, okay, that are within this, that are going to store the different parts, either the carbon, the water, the phosphorus, or the nitrogen. There's going to be something that stores it, and those are reservoirs. And then there's different processes that allow it to move between one to the other. I do not know to what level they're going to ask a question on this because we just got it back into the curriculum. And I don't even think it was in the curriculum when I started teaching 15 years ago. So like this is new to us. It was from apes. I feel like they brought it into us for some reason. Okay. So first thing is our hydrolytic cycle or hydrolytic. I don't know, whatever. Okay. Whatever. This is the water cycle. Okay. And so, yes, it will automatically record and post. So the hydrosphere is going to be the totality of all of the water that's on Earth. So we're looking at the oceans, the lakes, the rivers, the groundwater, the ice, atmospheric water, anything you see water, that is where is the hydrosphere. Okay, so the water is going to move between all the different parts and all those different reservoirs. Okay, so the main reservoirs that are straight out of the CED to know would be oceans, surface water, the atmosphere, and living organisms. Okay, so now I just found a random diagram online trying to show you the different processes that take place, but I tried to stick directly with what was in the CED to make sure that I was giving you everything that was in there. Okay, so now there are 4 different processes that we work with our water cycle. The first one's evaporation. I think you know evaporation. We've got liquid, and due to heat, that we break the bonds in between those water molecules that then cause our liquid to become a gas. Cool. Condensation is just the reverse of that. We now have this gas, it cools down, and because of the fact that it cools down, it goes from the gas to the liquid, it condenses, and now we'll have, of course, liquid. Okay. Precipitation is when we look at the like the liquid or solid water particles that come from the clouds. So like rain and sleet and snow, all those, okay, would be our precipitation. And then transpiration is evaporation specifically through our plants. And so due to cohesion and adhesion, the water that's in the ground comes up the capillaries of the plant and it ends up in the leaves. And so as the sun shines down, it's going to heat up that water, as well as there is a lower partial pressure of the water, the gas of water outside than it is on the inside. And so it causes the water to kind of evaporate and it comes out into the air. And that's what transpiration is. Okay. That's why you kind of talk about the stomata and you talk about those guard cells that are going to kind of help protect it to stop you from losing too much of your water. I don't know if they're going to give you a diagram for these type of questions or not. It's in the CED with those words, so I assume that they might not give you one. They might give you an FRQ over it. There's no way for us to know. So carbon cycle, this is looking at carbon moving through it, okay? So the big things that we see that has the carbon, we're looking at carbohydrates and carbon dioxide. There was an FRQ question in, I'm going to go with like 2012, 2013. It was a while ago, in which they had where you had to talk about the reservoirs for the carbon and how it moved from one to the other. And it was really just talking about photosynthesis and cellular respiration. Okay. But so this is how we can recycle the carbon. It goes between carbohydrates and then the carbon dioxide. Okay. And so photosynthesis is going to allow us to convert our inorganic carbon dioxide, and it then makes it into organic carbohydrates. Okay. You know all about that from Unit 3. And then cellular respiration is where we're going to convert those different organic carbon compounds. So like looking at our macromolecules, we break them down, our carbohydrates, our lipids, our proteins. We're going to break those down, and that allows us to then regenerate into inorganic carbon dioxide. Okay, as you remember, we release the carbon where in the whole process? Yes, we release it from the Krebs cycle. Awesome. So next thing we have is decomposition. So this is where we are going to break down dead and decaying substances, and you return them back into the inorganic. So like the carbon dioxide, we return it back to water. Simple sugars and the mineral salts are usually going to be a decomposer that does that. And if you hear my children, they just got home, so yay. So we have combustion. Combustion has to do with that we're going to have some type of reaction. So there's a fuel plus our oxygen, and that is then going to release energy, and it's going to generate heat and as well as fuel, and sometimes light. I'm sorry, we're going to break down the fuel. It releases heat and light. And so that's our carbon cycle that we have. So the nitrogen cycle, the largest reservoir we have for this is is the atmosphere. Okay. I'm going to be completely honest with you. I don't even know this cycle that well. I have to go back to this diagram every single time and memorize it. So at this point, I think you need to take these 5 words, and I think you just need to make yourself some note cards and memorize what these 5 processes are. Once you get what these 5 processes are, we should be able to answer any question that they have about it. Okay, so nitrogen fixation has to do with that we're going to fix the ammonia, and we're going to we fix the nitrogen to make the ammonia, and then that becomes ammonium once it gets the different hydrogens in the soil. Okay, nitrification is just how we're going to make the nitrate. Okay, and so we take the ammonia and we convert it into nitrate from that. Assimilation is when we can take the nitrate and the ammonia, and we convert it into the different organic compounds. So like proteins, amino acids, nucleic acids. So we assimilate it, we kind of make it into some type of structure, okay? Ammonification is when we convert the organic nitrogen to the ammonia. And then we have the denitrification where we're going to take the nitrate and then put it back into nitrogen. And so this is just looking at all those different processes. So you can see that we've got the bacteria, the denitrifying bacteria that's going to convert that nitrate. They make it back into the gas, as we said. So now it's back into the environment, right, into the atmosphere. That nitrogen, as we said before, we have our nitrogen fixing bacteria that will fix that nitrogen. They make it into the ammonia. They make it into the ammonium, okay? Then we see, of course, that ammonification, where we're going to convert that into the ammonia, okay? And so it's a lot of different moving around within this cycle. And again, if you can just memorize those words, I think you'll be okay. I don't know if you're going to have to memorize these definitions. These words are in the CED. This definition right here, or nitrogen fixation, is in the CED. The rest of them were ones that I had to look up or find. Okay, so the phosphorus cycle, this involves our weathering of our rocks. And so it's going to release that phosphate into the soil and the groundwater. And so producers are going to take up that phosphate and they're going to incorporate in their biological macromolecules. If you can remember, what are the two different places where we're going to see phosphate in the macromolecules? Yes, we're going to see them in the nucleic acids, as well as we're going to see them in phospholipids. And then the consumers are going to eat those producers to transfer the phosphate to the animals. And then the phosphorus is then going to get returned back by decomposition, and then incorporates back into the environment with the decomposition, as I said before. Okay, and so it's just a whole bunch of moving again. The phosphorus, so we got to just get it out of the rocks, and then it moves around. Okay, so on to back to the ecology that we're all used to. So exponential growth. Exponential growth is unlimited growth. So there is an unlimited amount of resources. So what we're going to see is that these are going to continuously grow. Okay. So the equation that's in our CED is R equals B minus C. Okay. Where R is our rate of increase, B is our birth rate, and D is our death rate. Okay. And so we'll use that R value, and then we're going to multiply that by N. N is going to be our population size. And I've got an example problem for us. But here you can kind of see what's happening, right? So our population size is exponentially growing. It just keeps going up and up and up and up forever, right? And so the example we have is if a population has 400 individuals and there's a rate of increase of 0.5, how many individuals will there be after 2 generations? So what we'll do is we'll sub it in, right? So R is going to be 0.5. N is going to be 400 because we have 400 individuals and our rate of increase was 0.5. So 0.5 times 400 gives me 200. And I'll take that 200 and add it to the 400 that was already there. So now after generation 1, I have 600 individuals, right? So if we wanted to do 2 generations, we'll go through it one more time. So we'll do 0.5 because our rate of increase is 0.5 and our population size is 600. So 0.5 times 600 gives me 300. And then that added back to the 600 gives me 900. And so after 2 generations, we went from 400 individuals to 900. So you can kind of see how it drastically increases. Versus logistic growth. Now, things cannot forever keep growing. They can't stay in exponential growth, okay? And so what's going to happen is there's this carrying capacity. And so the carrying capacity is the maximum organisms, individuals that the environment can sustain. And so now we have to bring in this K value. K is our carrying capacity. And yes, this formula and this formula are both on the formula sheet, okay? And so we're now hitting a carrying capacity. And so we're going to see that the population size is going to kind of level out right there, okay? So if we were to do the same example problem, but now we've just added a carrying capacity of 800 to it, we can see what goes on there. Okay, so 0.5 is going to be our rate of increase. 400 is going to be our population size that we started with. And then 800 is our carrying capacity. So 800 minus my 400 is going to give me 400. Divide by 800 gives me 0.5. So if I multiply 400 times 0.5 times 0.5, that gives me 100. And so I'll add that to the population of 400 that I had before. And so that gives me 500. So if I do this another generation, we now have 500 as our population size. So 800 minus 500, that's going to give me 93.75. And so my population size is now 593.75. Okay. What is the word after logistic? After logistic, it says logistic growth. The whole PowerPoint is posted online. I forget that I cover up part of my screen, and sometimes I cover up my words. I'm sorry. I just want to make sure everything was fitting on the slide. Okay, so density dependent factors. These are things that have to do with they're dependent on the density. Okay, the larger your density, the more heavily packed individuals there are in a small area. Okay, the more densely populated it is, there's going to be an increase in these factors, right? So competition. If you have more individuals, there's way more competition for that limited amount of resources. Predation. Hello, we've got way more prey. We're going to eat more. And then, of course, disease. Disease moves much quicker in a more densely populated area. Think about why we all get the flu during the winter time, but not during the summer. The flu doesn't go away. It's just that we're all inside and we're causing, of course, we're densely populated on the inside. And so it increases the rate of travel of that disease, which is the flu. So density independent factors has nothing to do with the density. Okay, these are just factors that are going to affect all individuals. It's going to affect them equally, no matter what. So natural disasters, or like an earthquake, a flood, okay, or human activity. These are all density independent. It doesn't matter how many individuals there are in that area. It's going to affect them the same. So community ecology. Now we have our Simpson's index. This looks at measuring the biodiversity, okay, the species composition, the diversity of it. And so we have this formula, 1 minus the summation. This summation, this little symbol right here, means I'm going to add together everything that's next to it, okay? Does she have rights? Checking. I think she does, okay. We see that N is going to be our total number of organisms of that particular species, and the capital N is going to be the total number of all the different species. But this will make a little bit more sense if we can look at an actual example. Okay, why did it go away? Here it is. So we have a sloth and a penguin. We made it really simple. We just have two different populations that we're looking at here. Okay, so there's 18 sloths, there's 13 penguins, but you're giving us a total of 31. So if we were to sub this in, right? So I have 1 minus, and then I'm going to 18. So there's 18 sloths, but there's a total of 31. So 18 over 31 squared plus 13 for the penguins divided by 31 squared. And so a lot of times the error I see the students make in this is that they forget to do the 1 minus when they're done with this. Yes, this is on the formula sheet. And so when we solve that, we then get 18 over 31 is going to give us 0.52, 58. The 13 over 31 gives me 0.42. Okay. And so then we'll square those. That gives us 1 minus 0.34 plus 0.18. So that gives us 1 minus 0.52. So that's 0.48. Okay. And so that's going to show us the diversity. The higher this number, the more diverse our population is. Okay. Now that diversity has to do with two things. The composition. Okay. So looking at how many of the different organisms that we have. So if we have 4 different species, that's our species richness, that's our diversity, right? And then looking at our composition, that would be the relative abundance. So how many of those 4 different ones do I have? Yes, this is in the CD. So the fewer components that we have in our ecosystem, the less resilient it is to change. So we need the population to be very diverse so that we can change if there's an issue that's happened into the environment, okay? The final review is actually tomorrow. So main diversity of the ecosystem that deals with our keystone species. So no matter how, abundant they are, they're going to have a very strong hold of the environment. Okay. The producers, as well as our different abiotic factors, so non-living factors, as well as our biotic factors, so those different living factors that we have. Okay. So as I said, the keystone species is disproportionate to their abundance. Okay. If you remove them from the ecosystem will often collapse. Every time I've seen an FRQ question on keystone species and they talk about removing that keystone species, the answer most of the time is that the ecosystem collapsed. You have to then expand and talk about why it collapsed. But usually part of the rubric or part of the scoring guideline is that the ecosystem collapsed. And so you would then have to say why. So here we have our sea otter. Okay, our sea otters are going to eat sea urchins, and the sea urchins eat the kelp. If for some reason we don't have any sea otters, right, then nothing keeps the sea urchins in check, and the sea urchins go crazy and they eat too much of the kelp, so we lose our kelp forest and all the stuff that comes with that kelp forest. Okay. So also looking at community ecology, how do these different groups of populations of different species, how are they going to interact with each other? And so we have a predator-prey interaction. Now, all these are going to be some type of positive or negative. And so I did it kind of showing over here, showing if it was positive or negative. So the predator is positively benefited and the prey is negatively benefited. Just so we're all clear, you do have to make sure that you can spell out what these mean. So the predator is going to eat the prey. Herbivory is the same idea, but instead of it eating another like animal, it's actually eating a plant. So herbivory would be your herbivore, which is positively benefited, eating a plant, which is negatively impacted, right? Competition is going to be negative, negative. There's only so much resources. And if they're both of our populations, there's two populations competing for that same resource, we're going to see that it's going to harm both of them because neither one of them really gets enough of the resources. We also have symbiosis. This has to do with parasitism. So parasitism, the parasite is benefited and the host is negatively impacted. Mutualism, they're both benefited. So like the sea anemone and the clownfish. Commensalism is a positive zero. So one of them is benefited and one of them has no effect. And so these different interactions can help how they're going to access different energy as well as the matter within that community. So looking back at our cooperation, so different ways that we can have cooperation, they're going to work together for a common goal or benefit. So the ones that we have would be mutualism, commensalism, and then of course altruistic behaviors will help with that cooperation. Trophic structure or trophic cascade. Okay, so we have a top-down control or a bottom-up control. The top-down control means that the predators control everything. So since the predators eat these different levels of prey, we're going to then be able to control the environment and maintain it. Versus the bottom up control means that if I control the resources that are in the producer level, then I can control everything that goes up from it because the producers lead to all the other higher trophic levels. Okay. And then niche partitioning, if there's only so many resources, then we're going to find that they're going to these ones that are competing are going to find a way to break up those resources in order to coexist in the same environment. So my textbook uses an anole. There's this little lizard type things. And so one of them will be on a fence. Some of them are in the higher parts of the tree. Some of them in the lower parts of the tree. Some of them are on the ground. And so since they've broken up that environment, and some of them hunt at night, some of them hunt during the day, since they've broken up their resources, they're now all able to coexist in that area. And so disruptions within our ecosystem. So this will be looking at the interaction that we have between the environment and the variation. This is kind of like a hodgepodge one. And so one example we have would be adaptation. So adaptation has to do with that due to there being genetic variation, there's going to be selective advantage, okay? Because of the fact that they are more favorable in this environment, they're going to be more likely to survive. And so we're going to see that they're going to adapt into their environment. So then heterozygote advantage has to do with that the heterozygote is going to have a higher fitness than the homozygotes, okay? So malaria is the big example of this, okay? So, not malaria, sorry, sickle cell disease. So if an individual has sickle cell trait, that means that they're a carrier, okay? They're not going to be affected as much by the sickle cell disease because only half of their red blood cells are sickle. And the rest of them, they can still make normal hemoglobin, they're good to go, right? And then the the malaria, the plasmodia, can't infect the sickled red blood cells. And so they're going to have a resistance from from the malaria. And so a heterozygote of somebody that has sickle cell disease that lives in an area where the malaria is a very high incidence actually has the advantage in that environment. And the mutations, they specifically said mutations are not directed by the environmental pressures. So you taking antibiotic does not make your bacteria antibiotic resistant, okay? I think that that's a misconception a lot of students have. That mutation already existed. There was a mutation in the population, and because we added antibiotics to the environment, those that had the trait to be resistant would be more likely to survive, and when we're going to see that we'll then have more of the population being resistant. I don't know of an FRQ question that's been on that. So invasive species, okay, these are also going to disrupt our ecosystem. They're unintentionally or intentionally introduced species. So the zebra mussels is an example of an unintentional. Unintentional would be the kudzu. We brought kudzu over, I believe, from Japan, and it was to help with the erosion. And so now it has taken off, and now everywhere you look, there's kudzu. And so the reason why these species are able to take off is because of the fact that they have no natural predators. They're introduced to a new niche where there are no predators. There's nothing competing with them. and they're able to outcompete their native species. So then we can see a loss of diversity because these organisms aren't surviving because the invasive species is taking over. Could heterozygote advantage be a type of incomplete dominance? Maybe if you're talking about like pleiotropy in terms of how one genetic trait can cause multiple different effects from it. So potentially in that aspect, incomplete dominance does have to do with different layers. So yeah, I could maybe call that a type of incomplete dominance. Okay, so human impact. So humans have a big impact on the disruptions that we see in the ecosystem. So they accelerate change at the local and the global levels to help different driving changes of the ecosystem, which then causes different extinctions. Okay. Biomagnification has to do with that if a lower level, let's say that there's, I don't know, some mercury in the water, and the mercury ends up in the shrimp. Okay. So then the organisms that eat the shrimp are going to get all the mercury that was in the diet of shrimp. And so whatever eats the fish, let's say the fish ate the shrimp, okay? Whatever eats the fish is then going to have an even higher. And so as we get to higher trophic levels, as we go up our trophic structure, we're going to see an increase in the amount of this like toxin, okay? And so we see biomagnification. Eutrophication has to do with our algal blooms, right? So there was a excess amount of nutrients that's been in the water, and due to those excess resources, we're going to see that the algae starts growing extensively, right? And so because it does that, it then depletes the oxygen because of as things die, we're breaking them down, we're using up all that oxygen for cellular respiration, and so it then takes rid of the oxygen, and then of course there is our eutrophic lake, and it's very pretty. Yay. We also have geological and meteorological events. Okay, so this is going to affect our habitat change and ecosystem distribution. Okay, and so different biogeochemical parts go between it. Ms. McClinton, can you take that question? I'm not really sure right now. So testosterone, oxyreductase in a liver enzyme, okay, regulates the testosterone levels in alligators. One study compared the oxyreductase activity between the male and female alligators from this Lake Woodruff. It's relatively a pristine environment, and Lake Acapulco, which is an environment that suffered from severe contamination, okay? And so here we see this one's pristine, this one's contaminated, okay? The graph above depicts the findings of that study. The data in the graph best supports which of the following claims, okay? So we're trying to see about the oxyreductase amount, and then we're looking at males versus females and the pristine and the contaminated environment. So environmental contamination elevates total testosterone activity in females, okay? So being contaminated, we're going to see an increase in the testosterone in the females. Or is it going to reduce the total testosterone in the females? Is it going to elevate the ones in the males? Or is it going to reduce the ones in the males? So those are the four different options of things that could happen, right? So here we see looking at the females. Okay, so when we go from the pristine to the contaminated, we see a reduction in the amount of our oxyreductase, right? And testosterone oxyreductase is the one that regulates the testosterone levels. Okay, if I look at the males, I see that these error bars overlap. Right, so you see that they have the same Y range, which means there's no statistical difference between the contaminated and the non-contaminated environments in terms of the males. Okay, so then that tells me that we're looking here that the females decreased. So of course we're looking at that the environmental contamination reduces the testosterone oxyreductase activity in the females. Okay, and again, these ones do not overlap, so I can say that it is statistically significant that we're going to see a decrease in that testosterone oxyreductase. You cannot say that the males had a difference because their error bars did overlap, okay? So, sand lances in the genus of whatever are small fish that function as keystone organisms in several coastal ecosystems. These sand lances are prey fish that support organisms at higher trophic levels. Ooh, they're prey fish. That's important. Scientists performed experiments to examine how sand lance populations are likely to be affected by rising temperatures and carbon dioxide levels associated with climate change. Okay. So sand lance embryos typically develop and mature into adult fish at low temperatures, 5 degrees Celsius, and stable low carbon dioxide levels, approximately 400 microatmospheres. Over the course of 2 years, the scientists measured the survival rate of the sand lance embryos allowed to develop and mature in the laboratory at 3 different, so we have 5, 7, and 10. And then we have different levels of 400, 1000, and 2100. Okay. Now I'm kind of showing you a strategy here. I highlighted a dependent and an independent variable differently. So you know that as you go through this, they're probably going to ask you your dependent and your independent variables. So if you have that little highlighter ready to go, you can highlight in your prompts. If you see them talking about that the scientists measured something, well, that's going to be your dependent variable. If they talk about the different things that the scientists did to it, they changed X, Y, Z. That would be the independent variable. Okay, and so that's kind of one of the strategies I'm trying to teach students is just use the highlighter a little bit more. So part A, describe the effect of increased biodiversity on the resilience of an ecosystem in a changing environment. Okay, describe the effect of increased biodiversity. Okay, on the resilience. So if I have more biodiversity, then what's going to happen in my environment? Well, I'm going to be resistant to that change, right? It's going to have a greater resilience. It's going to be able to survive better because it has an increased amount of biodiversity. We talked about this before. The biodiversity is going to allow for the resilience in that changing environment. So part B. And yes, if you want to answer these questions as I go, you're welcome to plop them in the chat as we go. Great job, Kaylee. So B, justify the scientists selecting 5 degrees Celsius as the lowest temperature and 400 microatmospheres as the lowest carbon dioxide level in their study. Okay. Well, we know that that is where they traditionally are going to be found. Okay. And the experiment is looking at how does increasing the temperature and increasing the carbon dioxide level, how does that affect it? Okay. And so it's going to act as a control because it's going to be the normal conditions. So we'll be able to see how does the increase in carbon dioxide affect it? How does the increasing of temperature affect it? And so these are the normal or the current conditions in which the embryos developed and were used as a basis of comparison. These current conditions were used as basis to compare the effects or the changes in the environmental conditions as we look at the increasing of our temperature and our carbon dioxide. Now, real ninja cooks, you can't just say it's control group because we're asking to justify, okay? So we got to give a little bit more into it of how, like, why is this my control? What is it doing as a control in this experiment, okay? So in part C, state a null hypothesis for the experiment. Null hypothesis. Oh gosh, what's a null hypothesis? So a null hypothesis is going to mean that the independent variable has no effect. Okay. So I always teach students to say this in the exact same way. The independent variable has no effect on the dependent variable. Okay. So we did here, right? We said the dependent variable was the survival rate of the sand lance embryos allowed to develop and mature. That's our dependent variable. And our independent variable was the temperatures or the carbon dioxide. Okay. So an increase, a change in temperature has no effect on the maturation of the sand lance embryos. A change in the carbon dioxide level has no effect on the sand lance embryo development. Like you had to bring it back to that, right? So climate change has no effect on the sand lance embryo survival, sand lance development, size of sand lance population. Increasing temperature, carbon dioxide levels has no effect on the embryo survival, sand lance development, size of sand lance population. There is no difference between the survival rates of the sand lance, the development, the size of it measured at all temperatures or at different temperatures, okay? And so that's all the different options. Keep in mind that anything that's underlined, okay, was expected to be in the response. So if you were aiming at the second one, you had to talk about temperature and you had to talk about the sand lance embryo survival or about the development or about the size of their population. Okay, part D. Scientists claim that the reduction in the population size of sand lances will affect the stability of the entire coastal ecosystem. Provide reasoning to support the scientists' claim. So, they told me the sand lances, I believe they said they were keystone species, right? They function as a keystone species, okay? So they're keystone, right? They're important. Why are they important? Why are we going to see a problem? Well, it says here, these sand lances are prey fish, okay? Well, if they're a prey fish and you reduce the sand lances, there's no sand lances, right? We decrease. There's not as many of them there, right? If there's less of them there, nothing can eat them, right? Because there's not really going to be a lot of them there. And if you have no food, are you going to survive? Probably not. Okay, and so this has a negative effect because of the fact that the other trophic levels depend on it as food. There's a negative effect because of the reduced amount of energy that's transferred. Okay, because they're eaten, we would have that energy that would go from one trophic level to the next trophic level. And so you can't just say because they're a prey species, because they're a prey species was directly in the prompt. Yes, I know they're a prey species, okay? But you had to talk about that they were being eaten, okay? That they were the food, that there was energy coming from it. Yes, they already told us that we're going to see it's going to affect the stability of it. You're right. We do see the ecosystem collapsing, okay? But it's because of the fact that there's no food. So beaked whales feed at various depths, but they defecate at the ocean surface. Nitrogen-rich whale feces deposited at the surface waters supply nutrients for algae that are eaten by surface-dwelling fish. Which of following best predicts what would happen if the whale population decreased? Okay, so I made a little drawing here. You can make little drawings on your little what's it called? Scratch paper. You can either write with words, you could write little drawings. A prey species means that it is a prey, it's being consumed by a predator. And so what we see here is that the whale is going to defecate, which then provides nutrients to my algae, and then my algae is eaten by a fish. Okay, that was my attempt at a drawing. Okay, so this is what would happen if the whale population decreased. So if I have less of the whale, so there's no more whale, bye bye whale, right? Then I don't have the feces. And if I don't have the feces, then my algae is not going to have any nutrients. And if my algae has no nutrients, then my fish has no food. And so that is the answer we're looking for, is something to do with the decreasing of our algae, the decreasing of our fish, something of that nature. Okay. Now notice I've not even read these answer choices yet. Okay. That is a strategy. Sometimes you can try to come up with the answer on your own. If you come up with the answer on your own and then you see it in the answer choices, there's a higher chance that that is the right answer. So A, there's a reduction in the surface nitrogen concentration, which would cause an algal bloom. Well, since I have less defecation, I'm not going to have as many nutrients, so there's not going to be an algal bloom. So A is out. The surface fish population will decline due to reduction of population of algae. Wait a minute, I said that. I said there wouldn't be as much algae because they didn't have enough food. And then, of course, my fish would decline because they didn't have enough food. Cool, I like B. Let's keep going. The remaining whales would accumulate mutations at a faster rate. Why would they get mutation? That doesn't make any sense. The remaining whales will be forced to forage at the deepest part of the ocean. Again, we're just looking at the defecation of them and how is that going to happen? What will happen because of that? So the answer, as we said, is B, and a lot of y'all said that. Y'all did a great job on that. Okay. So here is our practice problem for 2025, number 5. Following models represent all the interacting species of two different communities with some of the same species and feeding relationships. These models assume that both communities have the same initial biomass. The models can be used to understand the effects of human activities on the communities. Okay, so here we're seeing two different communities. Community A, you can see it's very diverse. There's a lot of different levels. And then community B, it's not very diverse. So part A, describe a characteristic of a community that makes a species invasive in that community, but not invasive in a different community. Okay. Now remember, part A is always going to ask you a biology question. So let's think back. What is an invasive species? An invasive species is something that was introduced into the area, whether it was intentionally or not intentionally, and it's going to have exponential growth because of the fact that it has no natural predators, it has no competitors, and it has an unlimited amount of resources. So if one community had a predator for it while the other one didn't have a predator, that would allow the invasive species to be able to survive in the one that didn't have the predator, right? Same thing with competition, same thing, all those. Okay, so there would be no or reduced amounts of natural predators in the species in the community where it's invasive. There'd be no or reduced numbers of competitors in where it's invasive. There would be no or reduced amount of diseases where it's susceptible, so where it's invasive. So those are the different options. They were looking at predators, competitors, as well as diseases. Diseases isn't in the statement in the CED anymore, but it could still be a fair game option. Okay, so part B, explain why removing PP1 will have a greater effect on community B than on community A. So, but PP1 is gone. Okay, well, PP1 is gone. PC1 has no food. That sucks. PC2. PC2 has less food, but it still has food. And then SC1, because this one probably dies. PC2 has decreased, so we might see a decrease in SC1 and then a decrease in TC1. But for the most part, we only lose PC1, right? Like PC1 is our only problem. But if PP1 over here goes away, there's no food for PC2. And then we're going to see a reduction in SC2 and a reduction of TC2, okay? And it's really not going to be able to bounce back because as you see, like this one's got multiple options of things it can eat. This one also has another option of something it can eat, okay? So it's not going to be as effective in community A as it is in B because B just literally everything dies afterward, okay? So in community B, there will be decrease in PC2, SC2, and TC2. In the community A, the PC2 has alternative food sources, as we said. There'll be fewer or less diverse primary producers, so there'll be fewer paths for energy to move through the community. So there'll be fewer species, fewer feeding interactions, or less diversity in B. So in the future, it'll be less resilient to environmental changes, okay? And then part C. Now, the format of the exam has changed a little bit since 2022. So we are in a hybrid model, and because we're in the hybrid model for the exam, you have a physical book that you're going to write your answers in, and you're going to see the questions on the computer. There are multiple versions of the exam, but they send the same book to everyone. And so because of that, we're no longer going to be annotating a diagram. And so now it's going to be more like a multiple choice question. So if you look at last year's exam question, it was like, choose which of these I think was an intermediate or was a product of the pathway, and you chose from 3 things that was in the pathway. The one that's in the CED is a cladogram, and you're like picking where on the cladogram would answer the question. They've like named each of them. Aw, thank you. So in terms of this one is going to be the old kind of one, but it's still like a good question, okay? So an invasive species that eats individuals of species SC2 is introduced into community B. Using the template in the space provided for your response for community B, indicate the feeding relationship for this invasive species by correctly placing I and V to represent the invasive species and an arrow to represent the feeding relationship within community B, okay? So a lot of y'all are answering, you're saying this next to TC2. You are correct. Because Vax is an invasive species and it eats SC2, we would just have to put it on the same level as TC2. I think a common mistake the students made on this is that they made it too high or too low, and it wasn't in line with the other. Okay, and then part D, explain how human activities that add toxins to the soil could change a community with many species in each trophic level, such as community A, into community with fewer species at each trophic level, such as community B. So how can we go from this community to that community? How can human activities do that? Well, as you add toxins to your soil, right, we're going to see that those producers are potentially going to die, right? We're not going to have as many of them. There's going to be a decrease in them, okay? And so what's going to end up happening is that we'll see a decrease in our producer level. Well, as we decrease some of these producers, they become more like community B. Yeah, we can have biomagnification where that toxin might go up the chain, okay? And so the options we have are activities to eliminate our primary producers, which reduces our species diversity at those higher levels, or they could cause biomagnification of those toxins, which reduces the species diversity at the higher trophic levels, okay? One second. Sorry, y'all. Okay, so the exam format. What does the exam look like? Okay, I mentioned a little bit of this a second ago. So you have a hybrid exam. All of your multiple choice portion will be on the computer. You will see the question on Blue Book. You will answer the question on Blue Book. Like if you have not downloaded Blue Book yet, you need to download Blue Book so that you have it to test out the system to be able to see what's going to happen, right? The second portion of your exam is going to be your free response portion, okay? Biomagnification is at the trophic levels. The toxin is going to build up in the higher trophic levels. So for the FRQ portion, you're going to see the questions in front of you, and then you're going to write out your response in your exam booklets. You'll just have this book. I have an example in a second. But you'll write your answers. So that's why it's hybrid, because the questions are on the computer, but you're physically writing your responses to the FRQs, okay? So section 1 is 60 multiple choice questions, and you have 90 minutes. So every 10 questions, check your time. Make sure that only 15 minutes has elapsed. If more than 15 minutes has elapsed, you're going too slow. I need you to speed up. If less than 15 minutes has elapsed, you're doing perfectly fine and keep on rolling, okay? Section 2, okay? This is where we have our 2 long FRQs and 4 short FRQs. There was some discussion earlier about how many points these were. So your long FRQs are going to add up to 18 points. Okay, so it can be an 8 and a 10 and an 8, or they could both be 9. Okay, so they do vary between those different numbers, but together they're always going to be 18 points. And then your short FRQs are all worth 4 points. Okay, and so you'll have 90 minutes for this. I recommend that you take about 5 minutes, you read through the prompts, you figure out which ones you want to answer, and then you answer in the order that you know. Okay, and you spend 20 minutes on the long FRQs, and you spend like 8 minutes on the short FRQs. On the multiple choice, you want to spend about a minute and a half for each, but some questions do take you longer, and some questions don't take you as long. So I just tell students that every 10 questions, look to see if 15 minutes has gone by, because that will give you an average of 1.5 minutes per question. Okay, so in terms of your blue book, okay, this is where you're going to write your answers. And then question 2 will be our graphing question. I can guarantee you that they've already told us the second question is a graphing question, okay? Someone says, how do I know the difference between a bar and a line graph? A bar graph is going to be categorical data. A line graph is going to be continuous data, okay? There's no partial credit when it comes to your FRQs, but each individual question is its own point. And so you don't have to get all of the points on that FRQ. Okay, so as a reminder, the exam is on May 4th. May the 4th be with us. And it is in 9 days. We are in single digits, everybody. Do not panic. So here's what the booklet looks like. So when you open it up, it's going to say begin question 1. You'll write your response here. It says do not skip lines. When they say do not skip lines, that means that they don't want you to skip every line. Okay. So they want, like, if you write a little paragraph and then you skip a line, and then you write a paragraph and then you skip a line, that is perfectly fine. Or if you do part A, skip a line, part B, skip a line, that is perfectly fine. Do not freak out. Okay. So then you go on to this next question. You're going to construct your graph. Please note that you can only use one question. These are scanned in and color does not show up on the scans. So all you have is a single color. Your graph should be done in pencil. That way you can erase and fix it if you make a mistake. We are human. We all make mistakes, okay? So you want to be able to erase if you need to erase, okay? Somebody says, what is the difference between a modified bar graph? A modified bar graph is not accepted, okay? I do not have it handy right now. I can try to, while I talk, bring it up. But the modified bar graph is going to actually have it's a line, like a dot, and then there's a line that comes out from both sides of it, okay? And so because of that line that comes out from both sides of it, that's like what you would show for like your error bar or whatever. But that's not accepted on the exam. Nope, I didn't find the year. So like there are some teachers out there who are telling their students to do it. Please do not be that student that does it. Here's a modified bar. So this is not accepted, okay? So you put the dot here for your mean, and then you put your error bars coming out from it. This is not accepted if this is a bar graph. If this is supposed to be a bar graph, it should look like a bar graph. Like there should be bars, and then your error bar comes out from your bar, okay? If it is a scatter plot, like this one is a scatter plot, then it's okay to draw it like that because this is a different type of graph, okay? This is a scatter plot. And so let me show you what the actual rubric looked like. So you can see that they number them 1, 2, 3, 4, 5, 6 because they're labeling their different individuals. Those of you that are now freaking out in the chat, this is what the data looked like. So it had 6 different strains of corns, and we were comparing how did the double strand breaks compared to the average number of crossovers. And so this would be a scatter plot. Sorry, I have to get back to the live stream. Okay, so back to what I was saying. So do not do a modified bar graph. Must be a real bar graph, okay? So when you're constructing your graph, use pencil. Do not do multiple colors, okay? And then here we have, of course, question 3, okay? So each question is on a different page. Now, you're in the groove. You're writing. You're answering the question. You're going. You're showing all this bio knowledge. You are being a penguin. You are doing success, and you are rocking this thing. You're like, yes, I got this, right? And then you flip the page, and it says, begin question 3, and you go, I already wrote question 3. I wrote question 3 on question 2, and you have that mini panic attack, and you take a deep breath. Deep breath. Calm down. It is okay. I promise. Okay. What I need you to do is you'll go to question 3, and you'll say, see question 2 for question 3 response. Okay. And your readers will be able to go back there and they will be able to look at your response in question 2. Okay, so please do not erase it, do not rewrite it, do not scramble, do not freak out. It is perfectly fine. I 100% promise you, your readers have access to your entire book, and they can go full book, and they can grade it on question 2. Okay, make their life easier and tell them where to go. They go full book no matter what if they don't see the response there, but this helps them ensure that they find your response. Okay, now somebody says, do you have to use black or blue pen? You are welcome to use black pen or dark blue pen, or you can use pencil on the exam. If you look at the very front cover, it can be pencil. It's perfectly fine to be pencil. Now, the scan looks better with a dark blue or a black pen. Okay, so that is why I recommend doing that. Do not use an erasable pen, because the erasable pen oftentimes can erase because it's due to, like, the way that they work is due to heat. So as, like, you use the friction and you erase it, it's the heat that's causing it to be erased. And so you don't want, while it's sitting in a truck, a hot truck moving around, that is going to go away. How do we know if a graph needs many lines, anyone? It's based on the data that you have, okay? Sometimes, yeah, it's Miss McClinton's right. Sometimes they do tell us what kind of graph, so don't mess that up, okay? But definitely use which one they tell you, okay? So overall tips and tricks for your FRQs, okay? You're going to read the question. I don't know if you heard me. I said read the question. Sometimes y'all get so into it and you're showing me all this great biology knowledge that you forget to answer the question, okay? Label your responses. A, B, C, D, okay? Make them all separate. It is not an outline if you do it that way, okay? It's literally just A, here are my sentences. B, here are my sentences, okay? And so when I say to do it in knowledge order, okay? So let's say that you know part C, right? I can look at C, I can tell you experimental design question. I'm gonna go ahead and answer that. So C, and I write out my answer. And then I'm like, ooh, I know A. So then I write A, and I write out my answer. And then I'm like, oh, I know D. D, write my answer. And I come back and I go B, I know my answer, okay? So you can write them out of order, okay? It's helpful if you tell us which when you're on, so like A, B, C, or D, but they're different questions, so we're not gonna be, confused which is which, right? Don't restate the question, okay? They've already asked you the question. You are wasting your time rewriting the thing back to them, okay? Now, if it's one of those identified questions and you're writing it into a sentence, you could write, the molecule is. You wouldn't write, the molecule that increased in the reaction that da-da-da-da-da-da-da was this. No, you just write, the molecule is, and then tell me what the molecule is, okay? Additional examples are not scored, okay? So denote what's your first example, what's your second example, what's your third example, okay? So like sometimes they'll say, identify 2 factors of natural selection. I don't know, I'm just making things up, okay? So, and you just start writing things, okay? So tell them the first example, the second example, and then that way they know what your 2 that you're trying to show them are, okay? Because if you just write it out, sometimes you accidentally fall into other points, and if you didn't correctly state them, you wouldn't get that point, okay? So beware of contradictions. Don't say, okay, well, the population increased in size. And then as you continue going, you're justifying, you say, the population decreased in size. You just contradicted yourself, and I can't give you any points if you contradict yourself, okay? Use diagrams. The diagrams can help you a lot, okay? Sometimes the answer is literally in the diagram and whether you can read it. The next live is tomorrow. Define your terms, okay? So don't just name drop. This had natural selection. Boom. No. Like, this had natural selection. The population was more favorable, and you go on through your explanation, okay? You want to write clearly and legibly. There are people reading your response, like actual people. I am one of them. They are sitting there at a computer for 8 hours a day, staring at a screen, watching, trying to read all the things that you said, right? And so if you write clearly, it's going to be a little bit easier for them to be able to find your answers and be able to give you all the points. If they can't read it, they're going to ask other people to help them read it. Don't get me wrong, okay? But if it's illegible and we're looking at it, we're doing context clues, we're doing everything we can, maybe we can't figure out what that word is, or maybe it doesn't make sense as on the rubric. Like, there's a chance that you might not get credit, okay? Now, yes, we will do everything in our power, 100% everything in our power. But there is you gotta make sure it's clear and legible, please. Cross out responses are not scored. You just do a single line through it. It's perfectly fine. Okay. Use your pen for all your FRQs, as I said before, but use your pencil for graph. Watch out for something that bleeds through, because if it bleeds through the paper, it's going to be on the back response, and then you can't read the back response either. And then watch your time. 5 minutes to read them and renumber them. 20 minutes for your long FRQs. 8 minutes for your shorts. Okay. Now, I did an activity with my students this past week. It was 2 weeks ago. And I went through all of the questions from 2021 to 2025. And I was pulling out all the science practice questions. And I was like, oh, let me do all the ones that are identify the control, identify independent variable, dependent variable, null hypothesis, just looking at a chart and analyzing the data. I pulled all those questions out. And so when I say these are our easy points, these are the things that you can get without knowing biology. These are things that you're applying information. You're just saying science practices. You're saying, independent variable. Can you read the prompt and pull out the important information? And so maybe a good strategy would be to do question 1, 3, and then 6, because that is where we saw those kind of questions in them. Okay. And again, you don't have to answer the whole thing, but maybe if you just go to those questions and you answer the null hypothesis, you answer the control, that's going to help you, because you can get those points right off the bat, right? It's a points game. The more points you get, the better off you are, right? So helpful resources that we have out there, right? AP Bio Penguins. Hi. Also AP Classroom. If your teacher has not opened them yet, please ask your teacher. Say, hey, can you please open up all the PPCs, the progress performance checks? Can you open those up and let me practice? Okay, because those are going to be the best questions you can find. Okay, AP Central practice FRQs, y'all. AP Central has them going only to 2023. Okay, if you go to my website, I have them going back even farther. I pulled all of them from the Wayback Machine, and I have all of them here for you. You can access any of them that you want going back very, very far. Okay. So they're all there. If you like to listen to stuff, the Absolute Recap does a podcast, and she has worksheets to go with it, but she does a podcast. So go walk your dog and listen to a podcast, go for a run, whatever you like to do, and listen to some biology, and I'll help you to review. Okay. Review book. My favorite is the Barron's book from Mary Worth. At this point, we are 9 days away. I don't think you're going to get the best out of a review book if you go and have to buy one. Okay. So see if you can borrow somebody's review book so you can kind of work through your weak sections. You probably don't have enough time to go through the entire book, but you can pick and choose your weaknesses and focus on those. Okay. So good luck on your exam. Here is the link that will help us get to our the Instagram stories. It'll get to my Instagram. And then here's all of the games for the Wayground, so the Wayground games. Okay. There's my website in case you want to use it. Okay. There was a big curriculum change in 2013. I only use questions I only really think 2013 to current are really the ones to use. The best ones are 2021 to 2025. So if you're running short on time, you're like, hey, which FRQ should I be practicing? Anything from 2021 to 2025 is the ones you should be using. But there are still some really good questions in 2013 through 2019. 2020 doesn't exist because that was the COVID year. So some of you keep asking, when is the next session? Hi, the next session is tomorrow. Hopefully I make the PowerPoint by then, because as of this recording at 1 p.m., I do not have the PowerPoint made at all. So we will be doing a cram session tomorrow at 3 o'clock. Okay. My goal is always an hour, but let's be honest, I do not stay within an hour. And if it's a cram session, who knows what's going to happen. So 2 years ago when I did my cram, it ended up being 2 hours, and I speed ran. I mean, it was bad. I speed ran. So I'm hopefully not going to speed run. But then there's also a 10-day study plan that you could be using where you just do a topic each day. But what questions do you have? And I can see the screen that I'm way behind. So I know you haven't gotten to the point where you're asking me any questions, but let me look through what questions you have asked so I can maybe answer those. For questions such as describe the function of ribosome, would one sentence be enough? If it's saying to describe, I'd probably do 2 sentences, maybe talk about, what the ribosome is, and then talk about what the function of the ribosome is. Do you think they'll switch up the type of graphs we use? There's a chance. It's been a bar graph for a while, I agree. But it could be any of those graphs that they have in the CED. The CED does have all those. I like how Ms. McClinton disagreed with me, but that's okay. We all have our own opinions, right? Let's see what else we got. Acceptable types of graph. What are the graphs you need to know? Write enough. Yes, I agree with Ms. McClinton. You got to write enough sentences to answer the question. So there's not really you must write 3 sentences. You must write 2 sentences. There needs to be something to it. Okay. So is it bad if you haven't started studying? Yeah, the exam is in 9 days. You should be doing something. I assume you've been working hard all school year and you've actually been studying for all of your unit exams. And so it's probably just going to be like a quick review for you. But you should at least kind of look over the CD, make sure you know all the information that's in the CD, practice at least minimum the 2025 FRQs. How can you improve on your FRQs? Literally sit down with a timer and write it. Um, and then look at the exemplars. Okay, somebody says, "Oh my God, what do you mean that there's other graphs?" So if you go to the CED, and you go to like the end of it, here. So they actually have in there the different types of graphs. So you see a bar graph, a histogram, a line graph, a log scale graph, which, yay, a double Y, a scatter plot, a box and whisker. Sorry, that's the box and whisker. Pie charts, and then they tell you how they're going to be scored. So they actually will tell you in here, this is how they're going to score the construction of it. That you have the appropriate type of graph for the data that's been plotted. You've appropriately labeled your axes showing the variable with the units. If you have more than one condition, that you've used some type of legend to tell them which line or bar represents each one. Your scaled intervals must be uniform, so make sure that you have a logical number. You can break your axis, but you want to make sure that there's nothing, graphed underneath it. The graph should clarify whether you start at 0, 0 or not. You can start at any number you want. Data plot in the graph should be easily identifiable. Error bar, so you want to make sure that it's not squished. Okay. Trend lines are appropriate when presenting the scatter plot. So that's kind of telling you how you need to have them. Somebody says you need to know how to draw all of those graphs. I don't know. You might have to be able to analyze them. I can't imagine you having to draw a box and whisker. You might be able to do that. Anyways, you don't have to label the type of graph because they're going to look at it and be able to know what kind of graph you made based on the graph you made. When we look at it, we can tell the difference between a line graph, there's a line, a bar graph, there are bars, a scatter plot, there is a trend line, and they're just the dots, right? So we can tell by looking at the graph what kind of graph it is. So I don't know if I've killed enough time. I hope that I'm there. I haven't seen the FRQ pop up, which makes me worried. Anyway, so I'm going to end this because I have to go to a family engagement. So I hope you guys all have a wonderful weekend. Um, Remember, AP Bio Penguins are just success. You're going to rock this exam in 9 days. I will be back online in 26 hours to review everything again with you. It's going to be a cram, and I mean like a cram. So